Welcome to A Conversation with History. I'm Harry Kreisler of the Institute of International Studies. Our guest today is Roy L. Caldwell, who is Professor of Integrative Biology at the University of California at Berkeley. Welcome, Roy.
Glad to be here.
Where were you born and raised?
I was born in Mount Vernon, Iowa, actually on a farm about 5 miles outside of Mount Vernon back in the 1943. Grew up on that farm, which was homesteaded by my great-grandfather, and went to the University of Iowa, both undergraduate and graduate degrees.
And how did your parents influence your character, do you think?
They wanted me not to be a farmer, and they wanted me to do something else.
So pushed you in the right direction.
Yeah. Yeah.
And how did rural life affect you, do you think, in retrospect? You obviously went to the ocean a lot, as we'll discover later.
I didn't go to the ocean at all.
Yeah.
I didn't see an ocean until I was 21.
Yeah.
I think it taught a certain self-sufficiency. Mm-hmm. You had to be a jack of all trades. It also gave me an opportunity to interact with nature a lot of hunting and fishing and just walking in the woods. An appreciation of animals and the life around me. I was lucky. There was a good school system nearby, and so I had good teachers. But I think probably the most important thing was just the opportunity to interact with nature a lot.
And when do you think you decided, hey, I think I want to be a scientist? Was it while you were still on the farm or later when you were in college?
No, it was I went to University of Iowa as an undergraduate. Actually, I think my first major was pre-law, and then Russian, and then pre-med, and somewhere along the line I discovered biology, and particularly zoology. There was one professor, Hugh Dingle, who was a new young faculty member, and he got me working in his lab, and I never really wanted to do anything else after that.
Before you were in college, did you read any books that you remember in retrospect that really affected you?
I think I read one autobiography of Madame Curie, and a lot of books on Vikings. Those are the ones I remember.
So that was your first introduction to the sea?
Probably, yeah. Yeah.
What is it that it takes to be a biological scientist? What are the skills you need?
Well, I mean, at least to work on whole animals like I do, animal behavior and ecology, it takes a sense of observation, a lot of patience, a lot of ability to tolerate frustration. Things just don't always go right. In fact, they usually don't go right. And you sort of muddle along until something does go right and seize it and work on it, and then it's back to the drawing boards and you sort of muddle along again some more. So there has to be an opportunism, patience, and you got to be a good observer. And I think you also have to respect your animals. You have to really understand them, try to understand them, want to know what it is that makes them tick.
In the biological sciences, I guess it's really important to focus on the part but also see the whole in many senses.
I think almost what you call a gestalt. If you look at just how the leg works or how the eye works or how the reproductive system works, very often you just don't have the context for how that system came into being, and you miss how it works in the overall context of the animal. So I think you have to know your animal, at least to some extent, even if you're going to work on the parts.
Is there, in biology, especially a sense of wonder that comes with the field as you contemplate, the ecology, say, of a particular animal?
There are times when you just say, how did this ever come to be? And then you stop and you think about it and you sort of go through the evolutionary steps of how we got from point A to point B to point C, and it falls into place. But you can't help but be in awe of how well the overall system works and how impervious to change it can be, but how rapidly it can change.
What about creativity in the biological sciences and the work that you do? You mentioned frustration. It must be the case that often you're on a path and your hypothesis proves entirely wrong, and then suddenly you sort of stumble into something new.
Yeah. You have to know when to cut your losses. There's no question about that. There are times when you are absolutely convinced you're right, and you know what the animal is going to do, and it won't do it. And at some point you have to say, no, we'll try something else. How you figure out what that other thing to try is something I still don't understand. But I think it helps, again, if you have a feeling for the animal, you can almost listen to that animal, and it'll sort of give you some hints as to how the system might be working. Where do I go next? If you just read the books, read the physics, the chemistry, very often you won't find the answer.
And so you come back to this point about observation.
Yeah, yeah. I think it's really important that you spend some time getting to know the beast before you start working on it, and it'll teach you a lot about itself.
Once you got into zoology and biology, did you expect to wind up in a focus on invertebrate marine biology, or did that surprise you?
That surprised me a lot.
Yeah.
I actually started out my PhD dissertation. I was looking at insect migration. I was looking at milkweed bugs in Iowa and how they found their way around. And then I just, as luck would have it, had a chance to spend a couple of summers in Bermuda looking at stomatopods, mantis shrimp. And then I went back to doing insect migration and actually looking at the hormonal control of it for a postdoc in England. And as luck would have it, Berkeley was looking for a marine invertebrate behaviorist. And I had all of 2 months experience, applied for the job, and I don't think they were very impressed with my marine invertebrate behavior, but I did know a lot about population ecology and physiology and genetics, and I got the job, and I sort of figured, well, I better become what they wanted, and so I've been doing marine invertebrate behavior ever since.
And I read a piece and in it you wrote of the growing satisfaction and occasional excitement of unraveling the biology of this creature. So tell us a little about this creature that you focus so much on, the stomatopod.
Well, stomatopods are marine crustacea. They all live in the sea. They're a very old group. They evolved probably about 400 million years ago. And they're characterized by what we call a raptorial appendage. It's a pair of arms that are folded up underneath of the head, and they strike out and catch their prey with them. They're analogous to the arms of a praying mantis, so that gives the stomatopods their common name of mantis shrimp. The difference is that the praying mantis strikes at a fly overhanded, like this, whereas the stomatopod strikes underhanded. Also, the strike of a praying mantis is about 100 milliseconds, about the amount of time it takes you to blink your eye. The strike of a mantis shrimp can be 2 milliseconds, 50 times faster. It's one of the fastest known animal movements. With the evolution of that appendage came a weapon system, a feeding system, and just about every aspect of the biology of this beast is wrapped up with that weapon system. And that's really what got me interested in them. Here's an animal which literally can kill its opponent with a single blow. So I first started working on communication and aggression, and now we're doing more work on reproduction and sensory systems. But it's all coupled with how do you deal with this armament and a very nasty lethal weapon system.
And there's a distinction that you make in your writing between spearers and smashers. Help us understand that.
It's a functional distinction. The first stomatopods probably made their living sort of sifting through the mud, sort of scrabbling, looking for things to eat, small animals. And probably what happened is that occasionally little creatures would be scared up and would be flushed. And the animals evolved a pair of elongated arms which could reach out and grab these fleeing prey. The first stomatopods had a barbed appendage which they used to reach out and spear or impale the prey and then pull it back in, subdue it, and eat it. Several times in the evolution of this group, that appendage has been changed into what I call a smashing appendage, or a hammer. And the way it works, let's say my hand is a series of spines. If I want to stab something soft like a shrimp, I can just stab it and pull it in. But let's say I've got something like a clam and I want to open it up. There's a lot of meat in there, but poking at it with barbs doesn't do any good. So one of the things the animals do is they fold this piece of the appendage back and they hit with the elbow. And that will break the clamshell. But if that becomes more enlarged and stronger, they can break stronger and stronger prey. Crabs, snails, big clams. And that gives them this really impressive weapon system, which is also very useful against other stomatopods. So the spearers are living on fish and shrimp and soft-bodied prey, and they're relying on speed to impale them and pull them in and eat them. The smashers go out, find something heavy, hard, break it up.
Now, having focused on this particular creature, having taken this job at Berkeley, you just told us, I'm curious, you mentioned that you began focusing on both the aggression and the communication system that went with that aggression. What in your background do you think led you to that particular interest or focus?
Well, I've been interested in weapons, for one thing. I was an archer and a fencer. The story of actually how I got into it goes back to a blizzard in Iowa. And I was working with my major professor, Hugh Dingle, and we were reading some papers about communication. And there was a new technique called information theory analysis, which had just come out to look at how much the behavior of one animal influences another. And it takes lots and lots of data for that to work. And Hugh had been in Bermuda as a graduate student and had seen these little animals that fought like crazy. And he sort of said, you know, these animals would be great for working on that. Well, as luck would have it, we were working on a cricket actually that day. And we came out about 3 in the morning. Out of the lab, looked out the window, and there was a foot of snow and 50-mile-an-hour winds, and we couldn't go anywhere. And we started talking more and more about this communication, and also how do you get out of Iowa in the middle of winter? And he said, you know, Bermuda's great, and these animals live there. And so we ended up writing a grant, and that's how I first started working on them. And it was to look at how information was exchanged between these animals, and I've basically been doing it ever since.
You said in one of your writings that it was studying the morphology, the behavior, and ecology of stomatopods has helped me understand the interdependence of each in the evolution of the whole. And I want to take that as a platform, because in a way here you have a very aggressive creature, and you became interested in that function and what it required in a way to actually deploy the weapon. Is that a fair?
Yeah. You usually in the evolution of a system can't change one component without affecting all of the others, and this is one of the prime examples of that. You add in a major weapon system, and now there's a need for information, good communication, good assessment capabilities. This weapon will dictate where the animals live, what kind of resources it uses, what it eats, every aspect of its biology. And so by looking at them, I never know whether I'm going to be working the next year on how they see and how they smell, or how they catch prey, or what have you. But it always comes back to this weapon and how it's evolved and what they do with it.
Mm-hmm. And you actually, in an earlier conversation, you were actually speaking in studying some of these animals, this animal and other creatures, that you can almost talk about arms races, basically. What do you mean by that?
Well, as the weapon increases in power, the animals have to deploy some sort of defenses against it, particularly if they're competing with others of their species. And you think of it this way, as the animal gets, let's say the animal evolves and becomes bigger and bigger. As it becomes bigger, the power of the strike will increase exponentially. It actually increases with the cross-sectional area of the muscle which powers the thing. Now, the major defense against that strike is the thickness of the body armor. Mm-hmm. Now, the thickness of the armor only increases two-dimensionally. It can get thicker, but if you have an animal and it grows larger and larger, the power of the strike will increase faster than the strength of the armor. And at some point, you have an animal which probably has a weapon which is capable of breaking down any armor that can be laid down. And that's happened several times in the evolution of this group. And one of the neat things about it is that in one of the groups of species where this has occurred, they have become very colorful, they use a lot of displays, and they rarely fight. They usually stand back at a distance and wave at each other and basically say, I'm bigger than you are, and please go away.
And we're talking about the stomatopods here.
Talking about stomatopods. Yeah. Now another, actually fairly closely related group has also gotten large, but it doesn't have bright colors, and it doesn't have a lot of displays. When two of them meet, they attack, and they go for a kill. Mm-hmm. So the question is, why? Why does one of them go in for all of this displaying and bluffing and sort of communication, and the other one just go in and attack? And it has to do with what they're fighting over. Mm-hmm. The one that displays digs burrows. Burrows are pretty cheap. It takes a few hours to dig one. And so it's probably not worth risking your life for a home. Mm-hmm. The other one, which is sort of the hawk, which goes for the kill, lives in pre-existing rock cavities, which are very scarce, and they have to fight for them. And so it's the value of the resource base in which they're competing which dictates how they settle their differences. And
so the focus on this one creature leads you actually, as you concentrate on it, leads you from its morphology, I guess one would say, to its ecology, basically.
And its behavior, yeah.
Yeah, and what? How do all biologists get focused on one particular animal, or are there variations? I mean, do some come in to biology and say, "Oh, I'm really interested in aggression"?
Yeah, those people who study animals, I always think there's kind of two types. There's the one that's interested in a particular function or a process. So they're interested in flight or aggression or game theory or something of that nature. And they try to pick animal models which will help them address those general problems. The other kind of biologist, which is my kind of biologist, is the one who works on one group of animals, gets to know that group very, very well, and then sort of allows the animal to pose the questions. And very often we don't know all of the latest technology and have all the specific knowledge of the problems. So then we collaborate with colleagues, and the two of us work together and try to address these issues. But I can't say that one is right over the other. If you work on systems and you go and pick an animal, you have to spend a lot of time learning how to take care of that animal, catch it, maintain it, do what you need to with it. On the other hand, if you work with an animal, you've got to figure out the problems. And very often, both types of biologists get together, as I do with several of my colleagues, and it's a very synergistic relationship that works quite well.
Now, before we move into another feature of this animal that you focused a lot, I want to do a segue here into where you go to study the animal. And as we're speaking in late June of 2001, you're about to go to Aquarius, and I want you to tell us what that is and how it empowers you in an important way to actually continue your studies of the stomatopod.
Sure. The Aquarius is an underwater habitat. She is anchored at 60 feet underneath the ocean on Conch Reef off of Key Largo, Florida. Right now, she's the only operational underwater habitat in the world where scientists can go and do research. She can house 6 people, 2 technicians and 4 scientists, and is about 50 feet long, about 9 feet in diameter. So we live in there for about 2 weeks. Mm-hmm. There's a hole in the bottom, and anytime you feel you want to go outside, you basically can swim out on Scuba and do your work. The advantage of this habitat is that because it is pressurized, mm-hmm. you're living at the same pressure that the water is at 60 feet. That means that we are totally saturated with nitrogen at that depth. Mm-hmm. And we can basically stay out indefinitely. Whereas if you're diving on Scuba from the surface, to go to a depth of 60 feet, you can only stay down for about 50 minutes before you would have to recompress or decompress on the way back to the surface.
Because if you rise too suddenly after you've accumulated all this
the nitrogen boils out of your blood,
and you have what is called the bends.
Right.
Right.
So literally, when you're in the Aquarius, if you had to come to the surface without decompression, you would probably be dead in a matter of minutes. Mm-hmm. So it's very much like a space capsule. Mm-hmm. As long as you stay down there, everything is fine. Mm-hmm. You just don't go up. But what it allows us to do is basically have much more bottom time to mm-hmm. work with the animals to conduct our experiments. So at what's called storage depth, at the depth of the habitat, I can literally stay out all day and all night if I want to. Mm-hmm. If I want to go to 120 feet, I can go down for a couple of hours, whereas if I was on Scuba, I could only go down for a few minutes. Mm-hmm. So it gives us a lot of time to plan and conduct experiments that we just absolutely couldn't do if we were working from the surface.
And you were explaining earlier that, for example, when you're just doing Scuba, it's not as if you go out and interview your creature. Actually, he's in a burrow, and you were explaining that that can be very time-consuming, finding him and so on.
Yeah, one of the things we were working on, I've done 2 previous missions on Aquarius, and the last time there's a species there that it turns out is monogamous, a male and a female live together in their burrow. Well, I never could really prove that because I never could get them out of the burrow. Mm-hmm. I was able to lie on the bottom for probably 6 hours, literally digging with fingers a hole about 2 meters in diameter until I finally got the whole burrow exposed and could prove that they were both in there. On Scuba, I never could have done it, or at the surface, I couldn't have done it.
So you're really empowered to truly embrace the ecology of this creature that you're doing.
Yeah, you're living there in its world. And we do a lot of work on vision and light. And it puts us into that environment. So we can measure the light, and we can actually see what these animals are seeing. And it's a very different experience than catching them and bringing them into the lab and then trying to recreate or simulate that environment.
Now, how long do you go down for in an Aquarius?
A typical mission is 10 days.
10 days. And is it claustrophobic? Is it... or are you so focused on your work that the time just passes?
It passes quickly, but I don't think anyone can stay that focused for 10 days nonstop. Mm-hmm. But the big advantage is every once in a while, if you just want to go outside, you can go outside mm-hmm. and go for a swim and look at some animals and sort of get away from these 5 other folks who are driving you crazy.
That's right. That's very good. And what has been the most difficult part of being on an Aquarius mission for you?
Occasionally, personal dynamics can be difficult, but I think it's actually just the general wear and tear on the body and the brain. You have always have an agenda which is 10 times longer than you could possibly accomplish. You're pushing from day one, you're aware that you've got 9 days left, 8 days left, 7 days, 1, you're done. So there's a lot of pressure. And the physical environment is demanding as well. That nitrogen that's stored in your body takes a toll. You're cold, you get tired, and you got those other people around, so the tempers get a little short. And, it's a physical and mental demand.
And it's a slow process of returning to the surface, right? Because your body has to adjust to the
It takes 23 hours, and basically what they do is the Aquarius stays put. She's attached to the bottom, and they just adjust the air pressure inside and gradually decrease the pressure until it eventually gets back to the surface. Then we go into an airlock. They repressurize us. We go out of the airlock and swim like the devil for the surface and come back to 1 atmosphere.
Now, before we go on to talk about vision, we've talked about the lab, the facility which you're able to work in, but one of the things that I noticed in looking, visiting your lab and actually some of the articles you gave me is photography has become an important tool for you in doing your work. Tell us a little about that.
Well, it's a way of seeing, a permanent way of seeing that's important to me. It's also a way of promoting my work, getting it well known. Pictures worth several thousand words quite often. When I first started, you know, I was no better than the average person with a brownie. I mean, I really didn't know anything about photography at all. I was very lucky early in my career to work with a number of filmmakers who became fascinated with stomatopods. And so
now, by filmmakers, you mean documentarians?
Yeah,
right.
Natural history photographers and filmmakers. And I worked with them, and just in the process of working with these people, I picked up some tricks of the trade. I also saw what keen observers they were, and they showed me a lot that I had never seen. They have a good eye, and that's why they're doing what they're doing. And so by using some of the tricks that they used, learning a little bit about composition, about continuity of shots, about how to control light, gradually my photography increased to the point where it actually became fun. And I knew I could take a picture when I wanted to, of what I wanted to take it.
And does that actually help you see the stomatopod?
Oh yeah, yeah. I can, very often I won't see something until actually I get the photograph back and I look at it and I'll see it, you know, doing something, or get the film back or the video back. This project that we're going to be working on the Aquarius next month, totally serendipitously came out of taking pictures. We had known for a long time that stomatopods could see polarized light. In fact, they can see 3 different E vectors of polarized light, much more than we can even with a pair of sunglasses. But we didn't know what they used it for. And there were ideas that maybe they used it for navigation or they used it to enhance contrast, but we just didn't know. And as luck would have it, a couple years ago, one of my former students and I caught a species of stomatopod in the Lembe Straits in northern Sulawesi. I brought them back to Berkeley, and they were gorgeous, colorful little animals, and I wanted to get some good photographs of them. Put one in a tank, and I was trying to get a photograph of it, and it had a light reflection, and I couldn't get rid of it. And so I grabbed a polarizing filter and put it over the camera lens and was looking and rotating the filter. And it's one of these times when you just can't believe what you're seeing. The front end of the animal has 2 large flaps, which are called antennal scales. And they were lighting up, red and purple and clear and white. And as I rotated it, just went back and forth. Here was an animal which had a polarized display. I don't know of any other animal that has that. As luck would have it, it's closely related to one of the species that lives underneath the Aquarius. And I had a few of those from our last mission. So I ran next door in the lab and grabbed a polarizing filter and looked at it. Same thing. Different pattern, but it was using polarized light as well. And so now we're going to go back and study how they actually use it and what they're doing with it.
Now let's go back to the morphology of this creature, because as one of the things that you wound up focusing at as you explored this creature, is, I guess we would say, the vision thing. That as part of the development of these tools for aggression, you were led to explore its capabilities for seeing. Tell us about that and the extraordinary complexity that this creature manifests.
Many of the animals, particularly the smashers, which have this dangerous weapon system, are extremely brightly colored. And they use those colors in a variety of displays where they threaten with their appendages and they jump up and down and flare things out to the side. And you can't look at them and not think, at least anthropomorphically, they're using these colored displays. Well, if they're using brightly colored displays, they probably are seeing color. So, as luck would have it, I was at a meeting in Italy several years ago, and I met another biologist, a guy named Tom Cronin, who worked on the sensory physiology of invertebrates, particularly crustaceans. And we started talking about stomatopods and color vision, and decided maybe we should look at that. And then we hooked up with another biologist, a guy named Justin Marshall from, at that time, Sussex. And the three of us started trying to figure out, did they see color? That's not an easy question to figure out, actually. We tried showing them colored targets and asking them to, can you tell the green square from the red square? They didn't always want to tell us the answer.
That's right.
And eventually, I worked out a technique that we took a little plastic cube and we drilled a hole in the center of it, and then we put some shrimp in that hole, and then we glued a piece of glass over the top of the hole. If the animal wanted to get the food, it had to break the glass, which it could do, and then pull it out. So it was like opening a snail. And then we put different pieces of colored plastic on the cube and would give the animal, say, a blue one and a green one. And it would be trained to open the green one for food. And they learned it very quickly. So that started to tell us they can see color. At the same time, Justin and Tom started looking at the morphology of the eye and the histology of the eye, and using some very sophisticated techniques, and discovered, first of all, that there were colored filters inside the eye. Now, not in all of the eye, just in 6 rows of visual elements that go down and bisect the eye. And these color filters were stacked up one on top of another. And it reminded us very much of some of the early satellites, remote sensing satellites, that could basically detect color on Earth by using a black and white Vidicon tube, and you take a picture through one filter, and then you take a picture through another filter, then you use a computer to sort of figure out the color image. They had that capability, it appeared. And so that was sort of our first idea of how they were doing it. Well, then we started looking at the visual pigments. We see color with 3 visual pigments, primary visual pigments. It turned out the stomatopods have up to 16 that have peaks of spectral sensitivity ranging from about 350 nanometers, well into the UV, way beyond what we can see, out to about 700. So they have tremendous capacity in terms of the visual pigments, and then you add on top of that the color filters, and that package has a mind-boggling capability of distinguishing spectral differences. Some of our most recent work has now shown that that system is tunable. Let's say we have an animal that lives from the surface down to 100 feet. At the surface, there's a broad spectrum of light ranging from UV out to red. And in fact, some of those filters go all the way from transmitting light in the red down to the blue. You take the same animal and put it at 100 feet, there's only basically blue light. And if it stays there for a few weeks, the filters change color and transmit different wavelengths. And they basically narrow or tune the light that's transmitted more towards the blue. It makes the eye much more sensitive to light, but it sees much less diversity of the spectrum. So it's a pretty complicated eye. As I mentioned, they can see polarized light. There's also the capability of incredible range-finding capacity. We basically tell how far something away is by parallax. They have two eyes, and sort of the images are slightly different. Stomatopod actually has 3 different focal points in one eye and 3 different focal points in the other eye. So it has hexnocular vision to tell how far away things are. So it's a very, very complicated eye that works very differently than the way ours does. Like I said, there's this row of ommatidia, or visual units, that come down over the eye. All of the color and the UV and the polarizing capacity are within that row. And so it scans the world by moving the eye stalk around and rotating it. So it's sort of like a scanner sort of looking at the world. And when it gets really interested in something, the 2 eyes come over together, and the rows actually cross, and it sort of looks at them.
So, and it's doing this line by line, like
it's basically a line by line.
Yeah. So in a way, we started with the morphology again. That is that the sight in part comes with this its armature and its function as an aggressor on the one hand, but it's also responding to its environment, right? In other words, because the ocean is a place where the range of light and so on must be quite extraordinary.
Yeah, I mean, from the surface, where the same light that we're seeing right now, by the time you get to 100, 120 feet, it's just blue. A lot of UV and polarized light in this environment, much less when you get deeper.
Now, in terms of the importance of vision for the animal's needs, I guess it relates to its observation and dealing with prey. It must also be related to getting food. It must also be related to mating, I guess, right?
Yeah, you have to be able to find a member of the opposite sex of your species. You have to be able to find animals that you are going to interact with. Have I beaten this guy up before, or can he beat me up? So there's information about your competitors. Is that fish a parrotfish, which is just going to munch on coral, or is that a jack, which is going to eat me? Is that a blue-ring octopus, which might eat me, or some just piece of algae? There are a lot of things that are color-coded. In fact, if you go into a reef in the Indo-Pacific, you may find 5 or 6 species of mantis shrimp, all living in the same habitat, using the same type of holes, and they'll be color-coded. One will have a big pink spot here, another one an orange, another one a blue, another one a white. So vision and color is very important to them. The question is, why is the eye so complex compared to, say, our eye, which we just have these 3 visual pigments that are sort of taking in 3 different spectra, and then our brain is taking that information and generating a color image, and we're figuring out depth and texture and all that other.
So a lot of complexity in the human brain for filtering the information.
You know, about 1/5 to 1/6 of our brain back here is involved in integrating, abstracting information from what comes from the eye. Stomatopod has a little tiny brain, actually, and apparently vision is very important for it. And so what it has done is process information peripherally by specific receptor types, and then that information is fed straight into the brain to be used, whereas we send sort of general information in and then process it centrally.
You mentioned earlier when we were talking that you came through experiment to the conclusion that these creatures had memory, basically, and that this was a system that could be useful in remembering a previous encounter. Tell us about that.
Yeah, it was one of these serendipitous discoveries that comes through the stupidity of an investigator. I was looking at aggressive behavior and fighting in these animals and trying to figure out what determined who would win. And in putting the animals together, one day I made a mistake and I put 2 animals together that had met previously about a week earlier. And they didn't fight. They just sort of looked at each other and went their separate ways.
But they had fought the previous
They had fought before. And I thought, wait a minute, maybe they knew one another and they remembered one another. So I designed a very simple experiment based on odor, and was able to very quickly show that they could identify the odor of previous opponent and remember whether they won or lost against that individual. And in fact, we later showed that mates can remember each other by odor for up to a month, and adjust their aggressive behavior accordingly. So they have a very long-term memory, true individual recognition, which is almost unheard of in invertebrates.
Now, let's go back to the Aquarius. You're going back there. This will be your third mission. On previous missions, you've studied the play of light in the ocean. What are you going to do this time? Let's focus on the vision part of your experiment.
Well, okay, this is the first all-stomatopod Aquarius mission, and we will be looking at vision, and as well as mating behavior and larval behavior. The visual component will be primarily looking at polarized signals. There is this one group of animals which apparently has a polarized signal, and they live there, fortunately. So we will be trying to figure out what they're doing with it. And we will be using models presented to animals in their natural environment. We will be altering the polarized signals to see how that affects the interactions among them. We'll also be measuring the transmission of those signals in that environment.
So observing to see what is going on. How are they using this information? What are they doing with it?
That's where it will start. We'll spend a lot of time just videotaping animals doing what they do. And then we will try to introduce other animals that have been manipulated, that either have enhanced or no polarized signals, and then see how the residents react to them.
Now, and when you've gone on one of these missions before, has something ever turned out an entirely different way, so you had to rethink how you wanted to do your experiment on the spot, or is that not easy to do once you're there?
Well, it's not easy to do once you're there, because very often you don't have the technology that you need. If it's just behavior observation, it's pretty easy to change your behavior. But if you need instrumentation, you can't just sort of call the surface and say send something down very easily. But certainly, on one of the missions, we just couldn't find any of the animals that we planned to work on. Fortunately, I went and found another one, a different species, and ended up doing some work on the evolution of monogamy. Otherwise, you're going to sit down there for 3-4 days twiddling your thumbs while you don't know what you're doing. You have to have contingencies, and we do have, like I said, always more work planned than you can do, and hopefully some of it will pan out.
And how long have you been planning for this forthcoming mission? Well,
I've actually started planning for this during the last one, when I started realizing that there were things I could do that I just couldn't do otherwise. But the grant proposal went in about a year ago, about 9 months ago, and we were told that we were going to go in December, so we've really been working now for about 6 months getting ready for it. And the next 2 or 3 weeks will be very hectic, getting all the material put together and getting everybody ready. And then there's a week of exhaustive training, and then a couple of days off, and then we saturate.
And I assume you have to be pretty physically fit to do this?
I'm running every day. Yes, you do.
And have you always liked swimming and going in the water?
I detest swimming.
I see.
It is a tool. It's not something I like doing. I like taking pictures underwater, but no, you would not find me swimming laps in a pool. It's not something I like doing.
So if students watch this interview and they say, hey, I think I would like to be a marine biologist, what advice would you give them to prepare for this kind of world and this kind of research?
Well, I didn't do a very good job of it. I started out trying to chase insects around the cornfield. I think you have to get a good, solid, basic education, and that starts with an overall basic education, and then in the sciences, physical sciences, good strong background. You have to know plants and animals, so you need some good diversity courses. Cultivate some of the tools that might be useful. In what I do, diving is important. A lot of people like to learn to dive, so if if you're going to be doing this for the rest of your life, you better enjoy it. And so some components of it had better be enjoyable to you. I like to travel. I like animals. I like diving. I don't like swimming, but I like diving. I like photography. So put together a set of tools that might be useful, a good basic knowledge, and then from there you're on a stochastic walk. And somewhere down the line, you'll probably end up doing what you like doing. I'm always amazed. Students apply to graduate school and they ask me, if they can come and work on sensory communication in stomatopods or something like that. And my first answer is usually no. Mm-hmm. And I don't even encourage them to want to be a marine biologist or an organismal animal behaviorist. It turns out, though, that if that's what the person really wants to do, and they're persistent, they usually end up doing it. Persistence is very, very important. So you have to enjoy it, you have to be persistent, you have to get some tools, good background, and then a little bit of luck, and you'll probably end up doing something that you enjoy.
Well, I'm curious as to whether you started as an environmentalist, as you've done marine biology, or have you become more of an environmentalist through the years as you've done this work, and if so, why?
I was not an environmentally concerned biologist up until a few years ago. Over the last 10 years or so, I've seen the pace of degradation of the environment accelerate to such an extent that it can no longer be ignored. Mm-hmm. I've gone places in the Indo-Pacific where for literally hundreds of miles you can't find an intact reef. I've watched the Florida Keys around Aquarius degrade over the last 10 years. It can no longer be ignored, and at this point it may be a bit late, but I have to add my voice to say we have to do something. And so I've become more aware and more active. But that awareness was late in coming, and it took some pretty rude observations to wake me up to what's happening to the world, and particularly to the oceans.
And what you're really talking about is going back to parts of the world a few years later, not having been there, not being there every day, and seeing the changes that have happened.
I just between the first Aquarius and the second Aquarius trip, it was obvious that the sea fans and the sponges and the corals were in much poorer condition than they had been just 5 years earlier. To go back Indonesia just 2 or 3 years later from one of our first trips and see an entire reef gone, just nothing left but pulverized coral. You can't ignore that.
And is this a sensibility that is common among the people who do the work that you're doing, and are they having a policy impact, do you think?
I think so now. I think 10 years ago there were not many marine biologists who were really calling for immediate action. I think it's the exceptional one now who isn't. We can't ignore it anymore. The oceans are warming, pollution's increasing, the environments are falling apart, and I'm not sure we can save them all. I think we can save some, and if we start soon enough.
Professor Caldwell, thank you very much for taking the time and taking on this fascinating intellectual journey and helping us understand how important the vision thing is in this little creature called the stomatopoda.
Stomatopoda, yes.
Stomatopoda, yes.
Enjoyed it. Thank you.
Thank you. Thank you very much. And thank you very much for joining us for this conversation with history.